Assalamu alaikum. Thank you for giving me an opportunity to share our research work with ICAZ 2021, which will be held in 24 and 25 November 2021. Organized by the Applied Zoological Society of Pakistan with the collaboration of ICAZ University, Islamabad. Uh, My name is Zafrulllah. I am from Pakistan University Department of Zoology, University of Balochistan, Quetta. Recently I am lecturer in University of Zafrullah, Balochistan. My research topic was development of fast digital method for the determination of pesticide chlorpyrifos in 5 local varieties of dry simple onions using fixture-based method and case chromatography mass spectrometric detection. Globally, onion is used both as cooked and uncooked due to its delicious flavor and widely used in all household items. There are many medical importance and significance of onion. Onion contains flavonoids and other compounds, and the sharpness of onion is the result of a volatile ion called allyl profile disulfide that play a significant role in preventing heart heat stroke disorder. The another disease like skin cancer, skin problem, diabetes, leishmaniasis, respiratory disorder, and many other local diseases are cured by onions. Onions are attacked by many several insects, several pests, and the most common insects and the most common pests that we see, Thalassophora, which are belong from Thalassophora, which cause a lot of damage to onion crop and cause a huge economic loss worldwide. The attack leaves become cold, wrinkled, and readily dry. The bulk is reduced approximately 60% as well as. From the 20th century, for posting of agriculture product, farmers use different types of pesticides like pyrethroids, termites, and organophosphate pesticides, which contain chlorpyrifos, which are used in the agricultural onion fields to control the insect pests. But if the pesticide like chlorpyrifos or carpamide or another pesticide, when the residues are remain in the foods, onions, or another such food particles, they are human, they are health hazardous. Uh, they are like act as a mutagenic, carcinogenic, teratogenic, genotoxic, as well as immunotoxin agents. The aim and objective of this study was to purify to percentage purify of chlorpyrifos pesticide through a high performance liquid chromatography to check whether the manufacturer company which provided the chlorpyrifos either the percentage is same or not. On the another hand, the chlorpyrifos residue was refined or checked in 5 local varieties by gas chromatography mass spectrometric detection using following the Christian methods. This is the experimental design. I use a randomized complete block designs. I use 3 consecutive sprays on onion when the threshold level of trips maximum up to 30 trips per day. Then I use a recommended dose 1 liter per acre of chlorpyrifos on onion field. I use 3 consecutive sprays of same concentration, and the food treatment was uncontrolled, untreated, which I compared the controlled treatment with uncontrolled treatments. Then finally, the 5 dry samples of onions, the 5 local varieties. The local name of the varieties is Chiltan 89, Pulkara, Trichmir, Nasarpuri, Gulnar. This is the local varieties, the local names of the onions which I have grown, which I have sown in fields. I have collected the random these 5 replications of 5 samples from the fields. Then analyze 15 grams of composition of sulfur containing onion, which the technique was given I will use. Then adding some chemicals. Then the method of the Christian method, the extraction is that which are shown in this slide. First of all, sample was homogenized, blended onion samples, Then analytical samples were applied. 15 gram of onion sample in 15 ml extraction tube was used. Then extraction adding some chemicals. Then vortex it for 2 minutes. Then centrifuge at 4,000 RMP. Then clean up it, collect the sample in tubes, and again centrifuge at 100 RMP at 6 minutes. Then evaporate some chemicals using rotatory evaporate reconstitution 1 ml in sample while and analyzed by gas chromatography. I used sample statistical analysis, graphic prism software were used. Then the data were presented as mean standard deviation. ANOVA difference will be considered significant at p is equal to 5, less than or greater than 5.5. The result of this study was when we applied the recommended dose of the chlorpyrifos, this the health significance, because if you use the recommended dose, there were no residue or remaining in the onion samples. I use the gas chromatograph. This is the chromatograph peak which indicates no residue in chicken 89 samples. This is the chromatograph of sample Pulkara. This is the chromatograph of Prichini, Gulnar, Nasarpur. This is the 5 chromatograph of these 5 varieties. There is no residue evidence in this. For the rectification of the fluorpyrifos technique, either they are rectified, they are corrected, they are validated or not. We spiked the Gulnar variety with chlorpyrifos when checked. Then at 9.66622 retention time, the graph was shown the chlorpyrifos residue. It means the system was validated, but there was no residue in the samples. Conclusion is that if we use the recommended, first of all, we use HPLC. We must be use HPLC to recognize to check the percentage purity of such pesticide, whether they are same or not. Secondly, we are use the recommended dose of the any pesticide in agriculture field to avoid the residues in any sample. If the residues are remaining in any samples, in any onion or vegetable sample, they are health hazardous. Therefore, we recommended our doses of 3 treatments with against the thrips on onion fields will be helpful for the research fields in these areas. So I am very thankful for your thank you for your attention. Wa alaikum.